Hi guys, welcome to Tidbits. So this year has been a crazy roller coaster one to say the very least, and that's still putting it nicely. With the global coronavirus cases topping 32 million, the vaccine for it cannot possibly come sooner. That being said, a lot also needs to be taken into account before one makes the decision to go for a vaccine whenever it's available. So I'm gonna walk you through all the different types of vaccine that are available and in the makes or in trial phases, the pros and cons of each one of them, so that you can make the decision whenever the time comes. Moving on.
So, the story so far is that the World Health Organization has listed about 180 COVID 19 vaccines that are being developed around the world. 48 of these vaccines are in the clinical trials on humans, and at least 89 are in the preclinical phase. 8 candidate vaccines are being developed in India, and to date, just 2 coronavirus vaccines have been approved. Sputnik 5, which was approved by the Ministry of Health of the Russian Federation and has raised significant concern and controversy about the vaccine safety and efficacy given that it had not entered the phase 3 clinical trials. And the second vaccine was also developed in Russia, Epivac Corona, which has been granted regulatory approval within the nation, also without entering phase 3 of the clinical trials. 3 Chinese vaccines have also been approved for use within China without phase 3 trials being completed. One of them has been given emergency use authorization in the UAE. Moving on to the topic of the vaccine itself. So each vaccine aims to use a slightly different approach to prepare your immune system to recognize and fight the SARS-CoV-2 virus that causes the COVID-19. However, we can group these technologies into 5 main types. Some technologies is tried and trusted, some technology has never been used before. And each of these 5 technologies is designed to prepare your immune system to recognize and respond to a future infection in a particular way. So the first technology would be the DNA RNA based. DNA and RNA vaccines use fragments of the genetic material made in the lab. These fragments code for a part of the virus, which in this case is the spike protein. After the vaccine is injected, your body uses instructions in the DNA and RNA material to make copies of the virus part or antigen. Your body recognizes these and mounts an immune response, ready to protect you the next time you encounter the virus. On to the pros and cons. The pros are that these vaccines can be quickly designed based on the genetic sequencing alone. Further, they can be easily manufactured, meaning they can potentially be produced for cheaper. Lastly, the DNA or RNA fragments do not cause COVID-19. And now on to the cons. There are no approved DNA or RNA vaccines for medical use in humans, as a result of which to face considerable regulatory hurdles before being approved for use. And as they allow only a fragment of the virus to be made, they may prompt a poor protective immune response, meaning multiple boosters may be needed. And then there's also the theoretical probability that the vaccine DNA can integrate into your very genome, which is a pretty scary possibility. So the speed at which these vaccines can be designed, needing only the genetic sequence of the virus, is why these vaccines were among the first to enter the clinical trial. Moderna, one of the most talked about COVID-19 vaccines, is an RNA-based vaccine that was advanced to clinical testing just 2 months after the virus was sequenced, meaning it can be a quite speedy process to develop a vaccine if one manages to get it right. The second category is the virus vectors vaccines. The infamous Oxford AstraZeneca vaccine is one of them, and so is the Sputnik 5. So the virus vector vaccines use a virus often weakened or incapable of causing disease itself to deliver a virus antigen into your body. The virus's ability to infect cells, express large amount of antigen, and in turn trigger a strong immune response, making these vaccines promising. Examples of vector-based virus vaccines include the vaccine made against smallpox and the one that is commonly taken for the common cold. The pros is that due to their highly specific delivery of antigens to the target cells and high expression of antigen after vaccination, often a single dose is enough to stimulate a long-term protection. But the cons, however, are that people may have existing levels of immune protection to the virus vector, reducing the effectiveness of the vaccine. In other words, the body raises an immune response to the vector rather than to the antigen. Next con is that the low-scale production of some of these virus vectored vaccines means they are less cost-effective. We have the third category, which is the inactivated vaccines. Inactivated vaccines are a tried and trusted method of vaccination. It's the technology used in the vaccine against polio virus and in some types of flu vaccines as well. Inactivated vaccines contain viruses treated with heat, chemicals, or radiations so that they cannot replicate but can still trigger an immune response. The pros are that it's a known technology, generally considered to be very safe. Well, not very safe, but safe enough. And another great thing about these types of vaccines is that it can be used in people with weakened immune systems. Cons, however, are that it has a low immunogenicity, so it requires multiple boosters. The fourth category is the live attenuated virus. Live attenuated virus are among the most successful existing vaccine strategies already used to protect against measles and polio. These contain virus weakened in the lab. The virus is still viable, that is live, but cannot cause the disease. After vaccination, the viruses in these vaccines grow and replicate, stimulating an excellent immune response. The pros are that they offer strong protection as the vaccine mimics the natural infection process. Moreover, it is also cost effective for large-scale manufacturing with a familiar regulatory approval pathway, meaning the entire process might also be more speedy. And then they offer single immunization without needing any extra dosages to stimulate the immune system. The cons are that it has a very rare potential, but still a potential to revert to a disease-causing state. Further, limited use in people with weakened immune systems due to the potential safety concerns. It can also require cold storage, which may limit the potential for distribution. We have the last category, which is the protein subunit. Subunit vaccines do not contain live components of the virus, but are made from purified pieces of the virus, that is, the protein antigens, and then trigger an immune response. Again, this is an existing technology. The pros are that with no live components, subunit vaccines are generally thought to be as safe, and it can also be used in people with weakened immune systems and other vulnerable populations. The cons are that the protein antigens that are to be used must be investigated in detail to ensure they elicit the best immune response, as insufficient immune response means that multiple boosters needs to be given. To summarize, in a nutshell, not all vaccines currently being developed to prevent COVID-19 will be successful. Safety issues or a lack of protection will halt some. So, a broad portfolio of vaccine approaches and technologies being progressed through human trials is quite reassuring. After all, we don't want to put all our eggs in one basket. Ultimately, it is likely that we'll need a repertoire of COVID-19 vaccines to offer widespread protection. Different vaccine formulations will ensure vaccination is safe and effective for all members of the society, including infants, the elderly, and people with weakened immune systems. I hope that this answers all your questions and maybe help make your decision whenever the time comes. Hope you have a nice day. Until next time.